Hi everyone, my name is Azrolan, and for the past 2 years I've been working on a research project called The Effect of High Glucose on APOL1 microRNA 193 in podocytes and its relevance in high glucose and puromyosin aminonucleoside or PAN induced apoptosis. I want to acknowledge Dr. Malhotra, Dr. Fishbane, Dr. Singhal, Mr. Despino, and Dr. Wazzi for all their help and support. In the United States, over 31 million people have chronic kidney disease, or more commonly known as CKD. CKD will ultimately lead to a person dying. It is suspected that too much sugar in your diet adversely affects the kidney and will cause apoptosis, illustrated in the top row. Apoptosis, which is programmed cell death in the kidney, could lead to CKD. The reasons regarding why patients develop chronic kidney disease remain unknown. A specific antibiotic called puromyosin aminonucleoside, or PAN for short, can also cause cell death, illustrated in the bottom row. A kidney cell is called a podocyte, and its structure is influenced by a gene called APOL1. The purpose of the study was to find the mechanism of podocyte injury linked to chronic kidney disease. High amounts of sugar intake in PAN can cause major damage to the cells of the kidney. Based on my background research, I first hypothesized that under conditions of high sugar intake, A. APOL1's production of protein will decrease, B. microRNA 193, which regulates protein production expression, will increase, and C. the structure of the podocyte will be disrupted. Additionally, I hypothesized that APOL1 protects against cell death in adverse environments of sugar and PAN through the decrease of miRNA 193A. I obtained underdeveloped podocytes from a special cell line from the University of Bristol, as depicted by the yellow cells. I allowed the podocytes to fully develop as a functioning kidney cell, which you can see by the middle row of the picture. Subsequently, I extracted the proteins using a Western blot technique, which detects and measures the amount of protein in the cell. The darker lines are the more expression of that protein there is, illustrated by the left-hand graph of the bottom row. I performed a densimetric analysis as well, which compares the standard expression of that protein to what was found in the experiment to see if there are any significant differences. The densimetric analysis uses the data of the Western blot to make a chart like the one that can be seen on the right-hand side of the bottom row picture. Each line of the Western blot represents a bar of the densimetric analysis. Then, podocytes with different levels of APOL1 protein production were subjected to high sugar content and PAN, illustrated by the sugar and PAN pointing towards the APOL1. Proteins were isolated from these tested cells and analyzed for the total protein expression of the apoptotic marker capsase-1, as can be seen in the picture illustrating the process above. I then found that podocytes displayed enhanced expression of APOL1, as you can see from the Western blot above, the darker lines signifying more expression of the protein, which is shown for APOL1. These findings support part A of my hypothesis. I then found that podocytes display enhanced markers of podocyte development, which are proteins called nephrin and CD2AP, and less expression of protein destruction of PAX2, as you can see from the darker lines of nephrin and CD2AP, signifying more expression while less expression of the protein called PAX2. However, turning off APOL1 and high sugar intake diminished podocyte development markers, as seen by the little to no expression of those proteins and more expression of PAX2. These findings support my part B and C of my hypothesis. Well, what can we conclude from this? Well, it can be concluded that APOL1 plays an essential role in the maintenance of the podocyte structure. If the protein production of APOL1 increases, miRNA 193 protein production decreases, resulting in podocyte development. However, high sugar content results in an increase in miRNA 193 protein production and a subsequent decrease in APOL1 protein production, which will lead to podocyte destruction. Such cell destruction leads to podocyte injury and the development of CKD. In addition to this, it can be concluded that there is a link that APOL1 protects against cell death in a high sugar environment, and in the presence of PAN through the decreased expression of miRNA 193A, supporting my second hypothesis. Thank you, and here are my references.